Our next speaker is Dr. Rachel Greenberg, who is at Duke Clinical Research Institute and part of the Pediatric Trials Network at Duke University. And Dr. Yao alluded to the Cuddle study. So Dr. Greenberg is going to speak on bridging the gap and understanding safety of off-patent medications in lactation, the Cuddle study. Thanks.
Okay, thank you so much. And thank you very much for the invitation and the opportunity to be here. I am Rachel Greenberg. I am a neonatologist by clinical training and a clinical researcher at Duke. And a lot of what I do as part of my clinical research work is with the Pediatric Trials Network. And I'll be talking specifically about the Cuddle study, but I thought I'd start out first by talking a little bit about the PTN. First, I have one disclosure, but not related to the content of the presentation. So what is the PTN? So you've heard little bits and pieces throughout yesterday and today about the PTN. PTN is funded through NICHD, exists because of the Best Pharmaceuticals for Children Act, and was really created to be an infrastructure for investigators to conduct the trials that improve pediatric labeling and ultimately child health. And the PTN exists because there really weren't the studies to inform drug dosing, safety, and efficacy in children for many years. Now there are some more legislation to ask pharmaceutical companies to do this work, but many of the drugs that are out there, most of the drugs that we use in children and infants are off patent and are used off label. And so how do we generate the data to inform that and also get that information into the labels so that it can filter down through the other methods of dissemination. So the PTN consists of a clinical coordinating center at DCRI, where I work. I oversee the day-to-day activities of the network in partnership with the PI of the clinical coordinating center, Danny Benjamin. And it's also, we also partner with the data coordinating center at EMIS. You heard Dr. Payne yesterday speak from EMIS, or other representatives from EMIS in the audience as well. And we also work with sites across the country to enroll participants. So you can see the network of sites who are active in PTN now. Of course, the participants and our partners at FDA and NIH. So just very, very quickly to give you an idea of the scope of what we do. So we were, the PTN was originally funded in 2010 and renewed in 2018. And since inception, we've enrolled over 12,500 participants in our studies, which have many of those studies were generated data that were submitted to FDA for consideration of label change. And we're very proud of the 21 different drugs and devices for which labels have been updated as a result of these studies. And importantly, I think we like to say we're agnostic to therapeutic areas. So whether it's cardiology or peds critical care, neonatology or infectious diseases or psychiatry, if the study is needed, if the data are needed, that's where we go. So this is just a list of the drugs and devices for which we've had updates. I highlighted the last two there, fluconazole and oxycodone, which were the most recent, occurring in 2024. And you'll hear more about oxycodone throughout this presentation. So I will move on now that you have a little bit more background about PTN to talking about the Cuddle study. So I think one of the best named studies that we have in terms of the acronym there, the commonly used drugs during lactation and infant exposure study. And I do want to take a minute to acknowledge Dr. Kevin Watt, who is one of our PTN investigators. He's the chief of the division of clinical pharmacology with within the Department of Pediatrics at the University of Utah. Many of these slides are his, and he, that he has led this study, but couldn't be here at this conference. So as the person representing PTN, I'm taking over as presenting to you all today. I think we all have this same background slide with these bullets, but I think just to again hit the point home, preaching to the choir, that women are and lactating individuals are basically forced to make a decision about whether to continue taking medically necessary drugs or to not breastfeed, or to how to make that decision without the data that is needed to make that decision. And so that's the motivation behind this study. And we've heard about LactMed today, so there are resources that exist. And even if you are a mom who is well informed and knows where to go, or has a provider that knows where to go to get information, it might go to LactMed to get information, for example, for a drug maybe like azithromycin. You may find some information there, but most of the drugs actually on LactMed don't have strong data. There was this publication 5 years old now, but I think still relevant at that time. Out of the about 1,400 drugs that were available in LactMed, only 2% had recommendations that were based on strong data. And so many of these drugs, the information that's available is not human data. It might be based on animal data or very small studies. So the Cuddle study was designed to try to bridge this knowledge gap. It was designed as a prospective, multi-center, open-label pharmacokinetics and safety study, and importantly is not interventional in the sense that the participants in the study are receiving the study drugs per standard of care. So they are people who are choosing with their providers to be on these drugs. And that is not part of the study. So we're it's an opportunistic in that sense that we're taking advantage of this off-label use that's already happening and trying to learn about it. So we've done this in other areas in pediatrics using the opportunistic model to do studies of multiple drugs at a time, and have had some success in generating data, with one study over many drugs. And that was the idea behind this study. So initially there were 10 drugs of interest, and I'll share a little bit later. We've added on to that, and a goal number of participants of at least 50 lactating individuals, and as well as their breastfed infants per drug of interest. We had more than 20 sites participating over time. The PIs are obstetricians, gynecologists, neonatologists, general pediatricians. So there are many stakeholders, providers that are interested in participating in this type of study. In this study, we include lactating individuals who are receiving at least one of the drugs of interest as part of their standard care, less than about 6 months postpartum, and their infants who are receiving at least some maternal breast milk. We obtain informed consent, and we only exclude individuals with known pregnancy during PK sampling. And so this is sort of a schematic to demonstrate what is done during the study. So moms participate. They happen to be taking the drugs of interest, and then they undergo inpatient or outpatient study visits in which breast milk samples are collected along with potentially maternal blood samples as well as infant blood samples. And then the goal is to kind of put all that information together and understand how the drug is passed from mom to mom's blood to mom's breast milk to baby. So this is an example of how a participant might participate in the study. So the participant might be enrolled and then undergo, that could be the same day, their first clinic visit. At that point, maybe they're enrolled because they were taking one drug of interest, in this case, metformin. At that first visit, they might have several samples taken, as well as a sample from their infant, and then mom's breast milk and blood. And then in between clinic visits, mom has the opportunity to report on events of special interest that might occur between visits and are also elicited at the visits. She comes back for a visit and she's still taking that same drug, so is able to provide another breast milk sample on that at that time. And then she may start on another drug of interest, and we can take advantage of the fact that some moms might be on multiple drugs of interest, and then at the next clinic visit be able to obtain data for both drugs. We also, as part of the study, collect data on the infants, any adverse events for the infants, as well as some long-term follow-up questionnaires related to development for the infants. So these were the initial 10 drugs of interest. As you can see, a range of therapeutic areas. So we've got antibiotics on there, antidepressants, antihypertensives, really commonly used drugs in the postpartum setting, including antiemetics and analgesic of oxycodone. So those were the initially enrolled ones. And in terms of how we decide which drugs to open, selection criteria include, there's got to be a knowledge gap. There's got to be, information that we don't know about that drug. That's not usually a hard to find because there's many drugs that have this knowledge gap. The frequency of use in the breastfeeding population is important. So because we're using this opportunistic design, we have to find drugs that are actively being used. And again, initially that's wasn't hard to find for the first 10. A lot of those are being commonly used. And then, also weighing the chance in terms of prioritization, the chance of the drugs actually potentially causing harm to the infant and might necessitate prioritization over others. So you can see we have a group, that first 10 that are now close to enrollment because they fully enrolled. We've got a group of drugs there now open for enrollment and a list that's growing of future drugs that we want to study. So since 2018, when the study started, we've enrolled over 1,200 participants. Here's the current enrollment for the currently open drugs. And for the ones that are fully enrolled, some of them have already been submitted to FDA with the great collaboration of the Department of Pediatrics and Maternal Health in helping to, because as these drugs are in so many different therapeutic areas, they will go to different review divisions, but we liaise with the Department of Pediatrics and Maternal Health to help that process along. So those 3 drugs have been submitted, and I'll talk just as a case example about oxycodone, since this is a drug that's commonly used, particularly postpartum in mothers who have undergone cesarean section. This is the old label prior to our study, showing there's the risk summary under the lactation section that there was a little bit of information note. Oxycodone is present in breast milk. There are variable concentrations. There's not a lot of specific data in the label to guide prescribing for mothers and providers. So it's this is the situation where we were before the Cuddle study. And so we were able to enroll 77 mothers and 73 infants in the Cuddle study. You can see the demographics here, and most were enrolled at within that first week postpartum. And just briefly, I'll present some of the results, and my other disclosure is that I'm not a clinical pharmacologist, so I present these results in a way that I understand them. A lot of you all know much more about clin pharm than I do. I've dabbled along the way in my role, but even I think I can understand these data, which show oxycodone concentration on the y-axis, time after last dose on the x-axis. And you can see here the triangles represent the concentrations in breast milk, which are sort of the highest in this group compared to the plasma concentration. So what does that tell us within moms? That oxycodone actually does concentrate in the breast milk. There's higher concentrations in breast milk than there are in mom's blood. But here, way down here, and this is a log scale on the y-axis, so you can see the infant plasma concentrations are much, much lower. and just kind of presented in another way. Here's now the milk to plasma concentration ratio on the y-axis at different doses. And you can see that there is the concentration ratio is greater than 1, kind of ranging between 2 to 4 in most cases. And that's for both commonly used doses of oxycodone. But despite that, the infant dose is low. We can see that its highest, the highest concentration is within the first 6 hours after dosing. But when we look at the average infant dose over 24 hours, and even the maximum dose over 24 hours that we saw in the study, it's quite low compared to the typical infant dose that's used to treat infants for pain, much lower. And then the relative infant dose is an important metric in lactation studies. So the relative infant dose here is 1.5% compared to the mom's dose, and generally accepted for safety is less than 10%. So the actual amount of drug that the infants are receiving in this for this drug and exposed to is very low. And so taking those that information together, in one slide, we kind of across many months and years of work to in collaboration with FDA to get to the point of a new label. And so this is what, if you look this up now, is available for oxycodone. So you can see under the risk summary some information about the number of infants and the number of mothers that were studied, what the relative infant dose is, and even some more data within the data section of the lactation section. So, this again is, I think, the package inserts, the labels are sort of the pinnacle. FDA is the gate for safety and in prescribing. And so when the information gets here, then, the next step is, I think, everything that it was talked about before in terms of dissemination of this information, and PTN is also working on best ways to do that. We have lay summaries, or what we call results at a glance, on our website, and are working to disseminate this information in the most appropriate ways. So just to wrap up for my last slide, this is just a summary of basically our pipeline of what I think is now becoming sort of a factory of study of drugs in lactation. So we've got the 1 all the way to label change, 2 are under review now and negotiation for new labels now. Another 7 are fully enrolled and are under preparation for data analysis and submission of the clinical study reports to FDA. And then another 12 are being actively enrolled. So hopefully this, pipeline will continue to add information to the community. And that is it. So happy to take any questions. I think that clarifying questions now and then a panel later. So if there's no clarifying questions, I'm happy to let the next person.
Can I ask one? How do you handle when the mom's taking 2 or 3? I think it's great you include them, but how do you handle it when she's taking 2 or 3 drugs?
In terms of the analysis,
yeah.
I think it's a great question. And depending, it really does depend on the drugs and the how they're processed. But for most of the, we do capture concomitant medications, and so those, depending on the number of moms, we may not have a sample, the sample size to fully evaluate a drug-drug interactions for. Within the 50 moms, maybe a couple are taking a particular combination, but that can be factored into the models depending on that the number that we have. But we do have that information.
Excuse me. Great talk, Sasha Ren from NICHD. Just a quick question about the Cuddle study biospecimen collected. So is there a plan, maybe I missed it, to share with the broad scientific communities, or has there already planned, implemented, or is there any example that outside of the investigator has requested to use that sample?
Yeah, so that's a great question. So our consent forms include consent for biospecimens to eventually go to DASH, and we heard about DASH yesterday. So as soon as we are finished with the analyses and the analyses that we need to do to get our drug concentrations. They go to a repository and then eventually are at DASH for access to other investigators, whether it's for these particular drugs or for other things that might come up. So yeah, that's our goal. It does often take some time to get to that stage. So I don't think at this point any of the breast milk samples are currently available for other investigator use because it just hasn't worked its way through that process yet.
Thank you. Thanks, Rachel.